Hey y'all, Dr. G here. So in this video we're going to focus in on the basic structure of nucleic acids. Okay, so what I'm showing here is a generic nucleic acid. Okay, and there are three important regions that I kind of want you to be able to recognize. Region number one here is our phosphates, right? So we note here that I have three phosphates shown. 1, 2, and 3. So this would be a nucleotide triphosphate. Okay. Region number 2, the central region, is our sugar region. Okay. And I'll point out here that I'm specifying an R group here because what's present in that position will dictate whether we have ribose or deoxyribose. And then in the last position, this is our base region, and this is where our nitrogenous bases are linked. Okay, so let's kind of think about some of the features of each of these. So first, in our phosphate region, I pointed out that here I'm showing 3 phosphates. Okay, and specifically, this is the alpha phosphate, the second one is the beta phosphate, and the third one is the gamma phosphate. So if you ever hear somebody talking about the alpha phosphate or the gamma phosphate, it just refers to the position in the triphosphate chain. Okay? Note here that this phosphate is always linked up with this carbon right here on our ribose or our deoxyribose. This is the 5' carbon. Okay? So the numbering system is fairly straightforward. 1' is always the carbon attached to the nitrogenous base. 2' is the carbon that determines whether we're dealing with ribose or deoxyribose. 3' carbon always has a hydroxyl attached to it. 4' is the carbon that kind of closes that circle. And then the 5' carbon is the one that's attached to our phosphates. Okay? This R group, if it's an OH, that corresponds to ribose. And this is the form of our ribose ring that we'll see is present in RNA and then a lot of other larger molecules that include nucleic acids as part of the structure. If this R group is a hydrogen, then this is a reduced ribose, and we specifically call it deoxyribose. Okay, as you probably know, deoxyribose is the form of ribose that's part of DNA. So this is important for DNA structure. Okay, we'll talk a little bit more about this in a later video, but the primary difference in the structure of RNA helices versus DNA helices boils down to the position here. When this is an OH, then the ribose ring has a different structure than if it's an H. Okay, and that very much impacts how the base group here is positioned when a double helix is formed. Okay, and the third place here is our base. Okay, this is always the 1' carbon attached to the nitrogen of a base. Okay, and these bases always adopt 2 primary structures, which we'll explore a little bit more in the next video. But for right now, I'll go ahead and kind of sketch out what this core structure is. Okay, we have 2 groups of nitrogenous bases. We have purines, which I'm sketching here. Okay, this is the core structure of a purine. I'm not going to put any attachments on it. This position right here is where ribose or deoxyribose is attached. This is the ring form of our purines, and our pyrimidines are a little bit easier. We only have a single ring structure, and our pyrimidines all have this core structure. Okay, so these are our purines, and these are our pyrimidines. Okay, the purines are adenosine and guanine, and our pyrimidines are going to be uracil, thymine, and cytosine. Okay, and we'll look at those structures in the next video.